We want to thank the organizers of the AACR for the opportunity to present on our NCI Consortia initiative, the response to the Cancer Moonshot recommendation to establish a network for direct patient engagement. The Blue Ribbon Panel Recommendation A called on us to enlist direct patient engagement through a federated network where patients will be offered comprehensive tumor profiling. The panel went on that patients are eager to provide their data and that efforts to reach minority and underserved populations will be a high priority. Several initiatives were launched in response to this recommendation, and each of these adheres to the principles of patient engagement, including ongoing and bidirectional and beneficial interactions between patients and researchers. We will hear both from researchers and patient advocates from these programs. This includes the Cancer Moonshot Biobank with the presenters of Helen Moore and Janet Freeman-Daily, the Pediatric and Adult Rare Tumor Network, or MyPART, Abby Sandler and Josh Summer, the Comprehensive Oncology Network evaluating rare central nervous system tumors, or NCI-CONNECT, includes Terry Armstrong and Kimberly Walgren, who will be presenting. And then on behalf of the Participant Engagement in Cancer Genome Sequencing Network, or PE-CGS, we'll hear from Nick Wagle and Maeve Smart. And with that, I'm going to turn it over to our speakers.
I'm Helen Moore from the National Cancer Institute. I'm presenting on the Cancer Moonshot Biobank today in collaboration with my colleague Janet Freeman-Daily. I have no financial relationships to disclose. Janet has a couple here listed. So the Cancer Moonshot Biobank is partnering with patients and NCORE sites to build a resource of longitudinal specimens and associated medical data to accelerate research on cancer drug resistance and sensitivity. This is NCI Clinical Study 10323, and it aligns with Moonshot goals in increasing patient engagement, increasing the diversity of research participants so that all can benefit, and to accelerate study on drug resistance and sensitivity. This study is enrolling patients with advanced cancers who are being treated with standard of care targeted therapies in the community. And our focus is on the collection of pre and post-treatment tissue and blood and data collected over time. This is illustrated graphically here. Again, we're working with NCI's Community Oncology Research Program sites to engage in the community for biospecimen donation over time. These specimens and associated data are processed through centralized functions, including a biobank, a data coordinating center, collaboration with our NCI patient-derived models repository, as well as centralized governance. And our goal is to get these specimens and data to cancer researchers, primarily for the overall goal of improving cancer treatment. A couple of unique aspects of this study are shown in these blue arrows. We are returning CLIA tumor characterization assays or biomarker assays directly to patients and their providers in this study. And we're also returning information about the study overall, or aggregate research results over time, to patients and participating medical institutions and investigators to try to be more transparent about what we're doing. The operational elements of the biobank are shown here, and some of those that are particularly important for the study are bolded. Those include electronic consent in English and Spanish, a participant and provider engagement portal, also in English and Spanish, a biorepository that is CLIA accredited and biorepository accreditation program accredited, and this is to enable our return of CLIA results to patients and physicians. We have a significant engagement component, And we will have an online catalog of available specimens for research. Our engagement strategy includes an external scientific panel composed of advocates, oncologists, clinical research associates, and more. And we also fund local engagement projects at participating sites. We develop newsletters and research updates and surveys as well, along with our patient and provider engagement website. There's a screenshot here from our patient engagement website. And you can see we have tried to really employ concepts around plain language here, trying to be very clear about what we request from patient participants, what we envision being done with the specimens, information about the biomarker tests that are available through the study, and so on. And you're welcome, more than welcome, to go and look at our website at moonshotbiobank.cancer.gov for more information. I'm also showing a screenshot from our annual reports going to sites to explain what we accomplished in the first year of our study, so through the end of 2021. How many participants were enrolled, how many biomarker assays were completed, and what's happening with the longitudinal collection that we're trying to achieve. And these reports go to sites with this overall information, but also information about how that site in particular contributed to the study. The members of our external scientific panel are shown here. And Janet Freeman-Daily, a patient advocate, is part of this panel, and I'm going to pass it to Janet now for her comments. Thank you all.
Hi, as Helen said, I'm Janet Freeman-Daily, and I serve as a patient research advocate on the ESP. I have metastatic lung cancer driven by an acquired rearrangement in the ROS1 gene. In my life before cancer, I was an aerospace systems engineer. I'm now co-founder of a nonprofit organization called the Ross Wonders, and I'm also a cancer research advocate who works with researchers, patient groups, regulators, medical societies, and nonprofits both in the USA and internationally. Cancer patient research advocates are people with close personal connections to cancer who are also passionate about helping research translate into meaningful outcomes for patients and families. We contribute to research by sharing the experiences, perspectives, and sentiments of patient communities with the research community. Our efforts can help in many ways: to define research questions that address patient needs, to improve protocol design to make clinical trials more accessible and more likely to complete accrual in a timely fashion, to co-author, review, and disseminate research articles, to help patients and caregivers understand the benefits and the process of research, and to develop mutually beneficial partnerships between patients, the community, and researchers. As a member of the ESP, I was asked to provide input on trial protocol, consent language, the e-consent process in the video, and the patient and provider portal. I learned a lot listening to other ESP members discussing their experiences, both with patient care and trial design, as well as the research process and the challenges of offering a trial across diverse cancer care centers. I especially enjoyed learning how specimens in the biobank might be used to generate more information for a variety of cancers. I believe I was able to bring a unique perspective to the discussions. Collecting biospecimens and conducting biomarker testing are areas in which I am very familiar as a patient and an advocate. The lung cancer patient community is at the forefront of precision medicine. Lung adenocarcinoma is increasingly being divided into smaller and smaller subsets based on driving oncogenes such as KRAS, EGFR, and ALK. My type of cancer, ROS1 positive lung cancer, occurs only in 1% of non-small cell lung cancer and has precious few biospecimens and cancer models available for researchers. Patients, clinicians, and researchers have shared with me their struggles with obtaining enough biopsied tissue to conduct biomarker testing, modifying workflows and staffing so that biomarker testing can be done reflexively, returning test results in a reasonable time period, and understanding the treatment implications of those results. I know lung cancer clinical trial participation does not accurately reflect the racial, ethnic, age, and financial status makeup of our community. In ESP discussions, I was able to share insights into patient sentiments, realities of patient care, language and terminology helpful in explaining the trial to patients, and barriers to trial participation. For instance, I connected the ESP and trial PIs with cancer patient nonprofits that were working collaboratively on standardizing the language around biomarkers. I also provided feedback to some patient perceptions of research biopsies. Metastatic cancer patients may be more willing to undergo research biopsies because it gives us some sense of control over a disease that has taken so much from us. However, before patients are willing to sacrifice convenience and comfort to give research biopsies, they must first trust the research process. There's a lot of work to be done to address diversity, inclusion, and equity in research. So Helen, you've been involved with a while for patient engagement. What are some of the important inputs that the ESP members provided to the study?
Well, Janet, the ESP's input was incredibly important and I guess surprising sometimes, which is really, I think, incredibly important. You mentioned the plain language input that we got and the input on, how to actually talk about a comprehensive tumor profile to a patient, so that it makes sense. And we adopted that language around biomarker report, biomarker test, biomarker report. We had great discussions and input on diverse representation. And the point that you made just now about the realities of patient care in the community, was really helpful to us around eligibility criteria, critical, actually. With an overall lesson that patients can have many different experiences when being treated for cancer. We need to be careful that we don't exclude patients based on our assumptions of how treatment regimens are implemented. So we thank you and your fellow ESP members so much for all of your input to make this a better study. So a question for you, Janet.
Okay.
So our group has been involved in developing best practices for biospecimens, biobanking, right, for years. And we have had excellent input from patient advocates over the years. But often, when we're reaching out for advocates, we find that sometimes they are pretty overtaxed. And my, a question for you really is, are there enough patient advocates involved in cancer research to really engage with new programs as they develop, because getting this involvement from the ground right through the conduct of the study is so important. Do we have enough advocates?
No. We definitely need more patient research advocates to meet the growing demand. And for a patient advocate to evolve into a patient research advocate, they need to learn more about the research process and the science behind treatments and clinical trials. They also need to develop communication skills that are effective with a variety of professionals. For instance, patients tend to communicate more about emotions, where researchers tend to communicate more about data, and they need to be able to talk to each other. Also, the patients have to be willing to represent the needs of patients whose experiences may differ from their own. But with adequate training, research advocates can make significant contributions to research in a variety of settings, such as the NCI spores, advisory boards for pharma firms, IRBs, grant reviews, or even FDA listening sessions. There's a growing number of programs to train research advocates and increase diversity in the patient advocacy population. The AACR Scientist Survivor Program trains advocates at key AACR meetings by bringing them to the meeting and engaging them in programs. The Research Advocacy Network offers training across cancer types. And then there are programs for specific cancer types, like the ISLC STARS program to train lung cancer patients that I'm involved in. There are many other programs available by cancer type and at some cancer research centers. So the listeners for this presentation can help by guiding patients who show an interest in learning more about their cancer and about the science of research to these programs, and by partnering with patient groups and nonprofits that aim to provide accurate and accessible medical information to patients and caregivers. So thank you, Helen, for allowing me to participate in the program, and thanks for doing this presentation with me.
Of course, and thank you, Janet, for all your contributions. It's really pretty amazing and great working with you.
Thank you.
Thank you.
Hello. Thank you to the organizers. I appreciate the opportunity to speak with you today about the My Pediatric and Adult Rare Tumor, or MyPART network at the NCI. This is a network that I co-lead along with Drs. Brigitte Wiedemann and Carleen Riley, and I'm excited to tell you a little bit today about what we're doing. I'll start by saying that I have no financial relationships to disclose. MyPART's mission is to increase patient and family involvement in rare tumor research and to develop new therapies for rare pediatric and adult solid tumors through an increased understanding of tumor biology and natural history. MyPART has a preclinical, a clinical, and an engagement component. MyPART is located within the National Cancer Institute in the Pediatric Oncology branch. The POB is one of over 40 labs and branches that make up the Center for Cancer Research, or CCR, which is one of the two divisions that make up the intramural program of the NCI. So the intramural arm of NCI is the arm that doesn't fund research grants. It's our in-house research that takes place on and around the main campus of the NIH in Bethesda, Maryland, or at the clinical center on the NIH campus, which is the world's largest research hospital. So MyPART is focusing on rare solid tumors that affect children, teens, and young adults diagnosed at age 39 or younger, although we do study all ages for tumors that occur across the lifespan. MyPART is engaging patients, family members, advocates, clinicians, scientists, healthcare providers as partners in our research on rare tumors. The centerpiece of MyPART is the natural history study of rare solid tumors, a longitudinal molecular, clinical, and patient-reported outcome data study that we've launched. Before the COVID pandemic, we were holding workshops and symposia on rare tumors to help develop expert consensus on research priorities and hosting multi-day clinics at the clinical center at NIH for rare tumors to bring patients and nationwide experts on specific rare tumors together. We're also building a network of sites outside of NIH where places can open the natural history study outside. As mentioned, the natural history study is a longitudinal evaluation, both retrospective and prospective. Enrollees can join either at the NIH Clinical Center or they can enroll remotely from their homes anywhere around the world. Patients, biological relatives, carriers of germline cancer predisposition mutations are all eligible to enroll in the study. Once enrolled, we carry out a number of clinical and laboratory evaluations and collect information on patient-reported outcomes and psychological, psychosocial functioning. We perform molecular profiling of tumor tissue, blood, and saliva. We have an annotated biospecimen repository, and we're working toward development of new therapies, endpoints for clinical trials, and screening guidelines. This is just a snapshot to show you how enrollment has been going on the natural history study that, of course, was affected by COVID. Before COVID, most of our enrollees participated from the NIH Clinical Center, but with COVID, we switched to mainly remote enrollment. Again, the advantage of the study, including an opportunity to enroll from home. The study opened to enrollment at the end of January 2019, and as of January 2022, over 420 participants have enrolled. The age distribution of the enrollees is shown on the left side of the slide, and that's age at enrollment. And on the right, you can see where these patients are coming from. They represent over 50 different tumor histologies, and they are located in 6 continents, 21 countries outside the US, and in the US in 45 states, including DC and Puerto Rico. As part of the effort with MyPART and the Natural History Study, we've established partnerships and are building relationships with 19 different rare tumor advocacy organizations. And we've done this to ensure, as we work, there is a two-way flow of information between us and the advocacy community so that they can share with us what's important to them, and we can share with them what we're doing and help them understand why the research we're doing is so important. An example of that is shown on the right of the slide. That's Dr. Mary Frances Wedekind-Malone, who's the PI of our natural history study, holding a session with the Chordoma Foundation with some of their patients to talk about the natural history study and in a very informal environment, help answer their questions and concerns that they may have about participating. And we found this forum to be very well received by our advocacy partners. That partnership with the Chordoma Foundation has developed quite strongly and is a really main reason why we've been able to enroll 60 people with chordoma for a tumor that occurs only about 300 times per year in the United States. But this has developed over time, starting with interest several years ago from several investigators in the intramural research program with an interest in chordoma, to a workshop that was held in the summer of 2018, where the Chordoma Foundation and researchers came together at NIH to talk about chordoma and research that needed to be done. As MyPART launched, the Chordoma Foundation became one of our first advocacy partners. And not long after that, we opened a sub-protocol of the natural history study that's specific for chordoma. So it asks a few more in-depth types of questions that are more specific to chordoma. We worked with the Chordoma Foundation hand in hand to develop the first pediatric chordoma clinic at the NIH Clinical Center. They were instrumental in helping us design and put the clinic together in terms of experts to invite to come for the clinic, and even help identify patients they were in contact with who would benefit most from coming to that clinic. And that in turn has led to multi-institution virtual tumor boards, pediatric chordoma working groups, with the ultimate goal of grant applications and new treatment trials for chordoma patients. And I'm happy to say we have a trial that we expect will be opening this summer that will be open for treatment of chordoma patients. I know you're going to hear more from our partners at the Chordoma Foundation, but if you're looking for more information, you can find out more about MyPART on our website, or email us, or follow us on Twitter. Thank you so much.
Hi, my name is Josh Summer. I am co-founder and executive director of the Chordoma Foundation, as well as a chordoma survivor. I'm very grateful for the opportunity to present today. I have no financial relationships to disclose. By a quick way of background, chordoma is a rare form of cancer that occurs in the bones of the skull and spine. It's primarily treated with surgery and/or high-dose radiation. However, with those therapies, there is a high rate of recurrence, and when the tumor recurs, there's oftentimes limited opportunities for a curative therapy, if at all. Consequently, the median survival for chordoma patients is about 8 years. However, there's a lot of variability. Some patients have a very rapid progression, while others have a more indolent course and can live years even with recurrent or advanced disease. Our mission at the Chordoma Foundation is to improve the lives of those affected by chordoma and to lead the search for a cure. And we do that by working to advance research to find better treatments, working to improve the care that's provided to chordoma patients, and providing a suite of services to provide education, navigation, and support to patients and caregivers at every step of their journey with this disease. There's a lot of research to be done, a lot of questions to be answered in order to improve care for chordoma patients, but particularly within the realm of clinical research, a lot of unanswered questions about therapies, for example, that are associated with better or worse outcomes, the range of expected outcomes for patients at various stages of the disease, biological correlates of outcome or response to therapy, as well as endpoints that are clinically meaningful. So for example, does tumor volume correlate with function or patient quality of life or overall survival? Answering these questions is really important for improving the care of chordoma patients, for identifying new hypotheses about treatments to be tested prospectively in clinical trials, identifying predictive or prognostic biomarkers, as well as developing more appropriate endpoints that perhaps could more sensitively detect benefits to chordoma patients. However, answering those questions is challenging for a number of reasons, some of which are idiosyncratic to chordoma, others are relevant to rare cancers generally. Chordoma presents in a number of anatomic locations in the skull base and spine, in adults and pediatric patients, and it presents at various stages of the disease. And in each of these instances, oftentimes chordoma is treated by different specialists, whether that be a neurosurgeon, an orthopedic surgeon, a neuro-oncologist, sarcoma oncologist, et cetera. Oftentimes patients will travel for treatment. Sometimes they'll have radiation at one center and surgery at another center. Their care will involve multiple different specialists. And so no single institution, no single specialist oftentimes has the entire clinical view of a patient's medical history. There's also a lot of variation across providers and across countries in terms of how care is delivered. There are a handful of centers that see high volumes of patients. However, even at these centers, it can be difficult to accrue a sufficient number of patients in various subpopulations. And the experience of those centers and the patients who are treated there is not necessarily representative of the patient population as a whole. So really, these factors conspire to necessitate a direct-to-patient research approach that can involve a cross-section of the entire chordoma patient population. And so to accomplish that, we've had as a long-term goal at the foundation to try to support or catalyze a national or international natural history study, and have been just very delighted and fortunate to be able to partner with the MyPART program on such an effort. There are a number of reasons why working with MyPART is so attractive to us. Number one, this is a institution and geographic agnostic protocol, so patients can participate regardless of where they were treated or where they live. There's a common infrastructure here that we are leveraging, not having to reinvent the wheel specifically for chordoma, yet at the same time, we're able to develop a chordoma-specific sub-protocol to ask and answer the questions that are particularly relevant to chordoma. We feel very, very fortunate to be able to work with the MyPART team, who has an extraordinary track record and can draw on the capabilities, the full complement of capabilities of the NCI, as well as that has a team that has the interest and the dedication to really make this project a success and see it through to completion. I think we're philosophically aligned around the importance of rapid data sharing. And we've been very heartened to observe how the MyPART team not only talks about collaborating with organizations like ours in the research and physician community, but really walks the walk and has embedded a collaborative approach really within the core of the way the MyPART program operates. So what does that collaboration look like? There are a number of different touch points and a number of different ways that the Chordoma Foundation has interfaced with the MyPART initiative. However, I think the key value and the key role that we've played is that of providing connectivity to physicians and clinical researchers, to basic and translational researchers, to key opinion leaders and expert physicians on our medical advisory board, to our community advisory board comprised of a diversity of patients and caregivers, and then to the patients and caregivers themselves via a number of different channels. In total, we're connected with about 90% of the US patient population, as well as quite a number of patients in Canada, Europe, and beyond. So this has really been an end-to-end collaboration from the very beginning, starting with working with the MyPART team to recruit a clinical working group to scope out and design the chordoma sub-protocol, working with that group as well as our medical advisory board to identify the key questions this protocol was going to answer, working with our medical advisory board to review case report forms to ensure that data is being collected in a way that is relevant to chordoma, is ultimately going to enable us to answer the questions we wish to answer. We've worked with the MyPART team to develop a number of educational pieces, including video, web content, print content, and had that reviewed by our community advisory board. We've worked to get the word out to patients through a number of different outreach efforts, emails, social media, patient navigation program, as well as our online community, et cetera. And finally, we've worked to get the word out to the physician community so that we can partner with physicians in helping to reach the accrual goals of this study and ultimately make it a success. So there are a lot of people to thank who have made this effort possible, certainly the MyPART team, as well as the Chordoma Foundation staff, the Chordoma Foundation's Medical Advisory Board and Community Advisory Board, referring physicians, and most importantly, patients and families who have participated in this study. Very much appreciate the opportunity to present today, and thank you for your attention.
Hello, I'm happy to be here today along with my friend and colleague, Kim Walgren. I have no financial relationship or disclosures. My name is Terry Armstrong, and I co-lead the NCI-CONNECT program with Dr. Mark Gilbert. We developed the NCI-CONNECT program, which stands for Comprehensive Oncology Network Evaluating Rare CNS Tumors, to address the unique challenges that patients with rare CNS cancers and their families face, as well as the researchers who study these rare diseases. I'd like to acknowledge our entire NCI-CONNECT team, our patients and their families, who have made it possible to develop this patient engagement network and a collaborative, well-funded research network for rare tumors, along with our advocacy partners. NCI-CONNECT is a program within the Rare Tumor Engagement Network, which is funded by the Cancer Moonshot and under a scientific priority designated by the Blue Ribbon Panel to establish a network for direct patient engagement. We studied 12 tumor types, each with less than 2,000 patients diagnosed each year in the United States. Our mission is to advance the understanding of adult rare central nervous system cancers by establishing and fostering patient advocacy provider partnerships and networks to improve the approaches to care and treatment. We have 3 goals, including developing a network of national and international institutions to study selected rare CNS tumors, to collect, analyze, and share data to enable a better understanding of the selected rare CNS tumors, and to develop standards of care. And then we hope to use these networks to investigate new therapies and methods to improve patient outcomes. As an overview of the areas we cover to engage patients, providers, and advocates in our program, we focus on clinical care and studies that can lead to new treatments, outreach and education that impact the person, and research and collaborations that advance our disease understanding. I'm going to talk briefly about our patient engagement through advocacy partnerships. We have 9 advocacy partners that work with us to identify and engage patients by disseminating information about clinical trials and new approaches to improve care and treatment. They help us share about our clinical trials and resources, partner with us to bring the patient and community voice to our workshops and meetings, and we've also partnered with these organizations to bring our expertise and resources to their conferences, meetings, events, and educational activities, including webinars. We've participated in about 7 events with our partners, including co-hosting the 4th Biennial World Summit of Brain Tumor Advocates, held at the NIH in partnership with the International Brain Tumor Alliance, a UK-based organization. We had 97 participants from 25 countries and 5 continents. They all participated in a 3-day meeting. In addition, we partnered with our advocacy organizations to bring educational content to the community, including a few examples shown here, a telehealth webinar with MBTS at the beginning of COVID in the University of Rochester, a medulloblastoma educational webinar with our expert Dr. Marta Pitas-Prado, and a national patient and caregiver conference chaired by Dr. Gilbert and myself and hosted by the American Brain Tumor Association. And as a last example not pictured here was a webinar on ependymoma classification with the CERN Foundation, which was live on their websites and our Facebook page at the same time. Our scientific workshops are an important area for advocacy partner participation. They integrate the patient perspective into these meetings with researchers, scientists, and other care providers to really make an impact on outcomes. We've held 7 workshops in the past 4 years, both in person and virtually. And for our virtual meetings, we've averaged over 300 registrants per workshop. Some examples are that we've held an outcomes assessment workshop in person, and then in 2020 held a rare CNS tumor virtual symposium in which we brought together international experts for educational presentations, a QA session, and panel discussions, with over 500 people registered for the 4-hour webinar. A partnership event with the Society for Neuro-Oncology in June of 2021 was a virtual symposium on survivorship care. There was first day's educational lectures with nearly 200 participants, including patients and their caregivers, and a panel discussion and second day focus on working groups in which we determined action items going forward to improve survivorship care. They have the voice that really brings the patient's perspective to these important scientific meetings, so we engage together. Here's some other examples of the way the partners impact these meetings through being members of panel discussions, moderators, presenters, and even when we held a virtual workshop, they submitted videos to share their stories and their messages of hope. At our global research in rare CNS tumors, where we were able to get all 9 of our advocates to record video questions to ask our experts, we also hosted a Facebook watch party to share with our community, and partners shared videos of personal experiences. At our survivorship symposium, all 9 partners provided videos of hope, participated in panel discussions, and small working groups. At our workshop on oligodendroglioma, Brock Green, who leads an advocacy organization devoted to oligodendroglioma, shared his personal story. And at our medulloblastoma workshop, Nicole presented their support groups and how they reach out to both patients and caregivers. Results of these partnerships is highlighted here with our medulloblastoma workshop, where we published a proceedings paper, which is the standard approach that we do. And because of this and our conversations at the workshop, we're able to suggest and lead to changes in NCCN guidelines for the care of adult patients with these diseases, directly impacting how patients are cared for. Another indication of the value of these partnerships is through clinical studies and trial participation. We have 10 active clinical trials and have accrued 563 rare tumor patients. Here we've highlighted 2 of these natural history studies, one that is totally web-based and one where people come to the clinic. And our advocacy partners refer patients for these trials in which we're trying to understand the disease and impact on the person. I also wanted to quickly highlight how we've created more awareness for our program through our website, social media accounts, and e-newsletter. We share educational content on these platforms, which our partners can syndicate or use on their website and share as well, helping us to maximize our reach to patients and their families. We're also focused on how we can reach diverse and a broader audience. And in March in 2020, we launched a Spanish language version of our website. In its first month, it reached 165 visits, and now, as of December of 2021, it receives over 15,000 monthly visits. This means it's seen a 7,329% increase in monthly visits in 1 year. We think this identifies that this was an unmet need, and we're providing a valuable resource to this population. In summary, I hope I've shown you that patient advocacy groups are integrated into patient communities, and by forming formal relationships that are mutually beneficial, we've shown here that through our event collaborations and partnership, we can successfully reach and engage more patients. Now our partner and my friend, Kim Walgren, Executive Director of the CERN Foundation, a program of the National Brain Tumor Society, will share her perspective of the benefits of our partnership to reach patients. Thank you.
Hello, my name is Kim Walgren, and I'm the Executive Director of the CERN Foundation, a program of the National Brain Tumor Society. And it is a privilege today to speak on behalf of the NCI-CONNECT partnership that we have with the NIH. It is a necessary partnership, and I can speak from the patient family perspective in that we talk with ependymoma patients and care partners on a daily basis. The partnership with NCI-CONNECT allows us to provide access to important information. Rare diseases present many challenges, as you are all aware, but access to quality data that is meaningful is extremely difficult, and it is an unmet need of our underserved community. I believe the word partnership is so important here because it reflects that we both bring value to the table. And I think that by partnering with networks and advocacy groups like the National Brain Tumor Society, we bring the humanity of the disease to the table. And so it's not just statistics, it's not just data. These are humans, these are our stories, this is our life. And so we try to work together to interpret this data and bring it to the community together as a solid front, as a united group. And hopefully that data can influence the personal experience with the disease in a positive way, even if it's hard information to understand or hard to accept. That is not something that we're afraid of. It's just something we don't even have access to. We have a very large unmet need of having information available to us that helps us to understand about what might happen with our disease. What will our disease do to our life? That is currently a very large unmet need, even though we have been working together in partnership for years now. We have to continue to unveil those issues that are most important, meanwhile focusing on paths to finding treatment. It really is a dual parallel path that must be considered in tandem. Living with rare disease is extremely difficult, but the lack of information and resources for support adds to the challenge. We would not be able to deliver the high quality programming that we currently are able to because of our partnership with the NCI-CONNECT program. That program equips us to be a trustworthy site with latest information and also the latest support of experts who understand this disease beyond a general setting. We're able to guide and support, to elevate, and to encourage people to take that next step, no matter where they're at, because we believe there's always an option for hope. There's always an option to be involved in your care, even when facing extremely difficult decisions. So many times I find that groups are duplicating efforts, and we really don't have time or resources to do that. And so I believe that partnerships are a wonderful way to celebrate the different gifts that each organization has and come to the table and use those together to propel this community forward. I truly believe that institutions operating alone with only laboratory science and data will never meet the true needs of people and families going through what is most likely the most difficult experience of their lifetime. This rare disease, ependymoma, is not limited to the United States. So truly we are talking about an international approach to uniting and carrying forward our voices, our experience, our stories, and our outcomes. That by demonstrating a unique collaborative effort, we are also providing assistance and access to people around the world. And that is an incredible opportunity. And it means a great deal to anyone that has been on the receiving end. All you would have to do is talk with them or ask. And I guarantee you will be affirmed in your belief that these efforts and these partnerships are tremendous value. Thank you.
Hello, my name is Nick Hilwagle from the Dana-Farber Cancer Institute, and today I'll be talking about participant engagement and cancer genome sequencing network. These are my disclosures. The goals of the PE-CGS network are to promote and support the direct engagement of cancer patients and post-treatment cancer survivors as participants in cancer research, and to use such approaches for rigorous cancer genome sequencing programs, addressing important knowledge gaps in the genomic characterizations of tumors in the following areas: rare cancers or rare cancer subsets, highly lethal cancers, cancers with an early age of onset, cancers with high disparities in incidence and or mortality, and or cancers in understudied populations. As part of the PE-CGS network, there have now been 5 centers funded by the NCI, as well as a coordinating center. And they're shown here. Each research center engages diverse communities to generate scientific insights that could be applied to the development of new cancer therapies, improvements in the methods of cancer diagnosis, or identification of opportunities for cancer prevention. And these centers are the Broad Institute Research Center, the Washington University Research Center, the University of Southern California Research Center, the Yale University Research Center, and the University of New Mexico and TGen Research Centers, with a coordinating center at the Ohio State University. Listed here are the specific projects addressed by each of these research centers. You can see that there is a diverse array of cancers that these projects focused on, as well as a range of population focus and a variety of genomic characterizations. By design, all of the projects focus on whole exome sequencing, whole genome sequencing, and RNA sequencing of tumor material as well as germline material. And individual projects may have different additional genomic characterizations that are doing based on the specific questions that they're trying to answer. You can see that the cancer focus in the Count Me In project is leiomyosarcoma and osteosarcoma, and I'll talk more about that project in a little bit. In the University of New Mexico TGen Mayo Clinic project, they're focused on cancers with disparities, particularly in GI and GU, amongst American Indians of Southwest tribal nations. In the Optimum study, they focus on low-grade glioma in young to middle-aged adults. In the USC study called COPEC, they focus on colorectal cancer in Hispanic Latino population. And in the Washington University PE-CGS, they focus on multiple myeloma, cholangiocarcinoma, and colorectal cancer amongst Black Americans and any adults with cholangiocarcinoma. And when you look across the specific areas that are to be addressed, the knowledge gaps are to be addressed, you can see that these projects as a whole cover many of them. But importantly, all of them focus on understudied populations. Now, each one of these projects is going to focus on their own questions and are going to be important to fill the knowledge gap in that particular area. But the other important aspect of PE-CGS is that the network functions as a collaborative network to address common issues, share best practices and lessons learned, and utilize common methods when appropriate. And we now have been a network for about a year. And some of the areas that we have focused on as a network include determining best practices for direct participant engagement in cancer genomic research. And this includes things like learning and responding to patient participant preferences and identifying optimal methods of engagement and communication, studying key issues of disparities and equity in cancer genomics research, addressing gaps in molecular profiles of cancer, and developing a shareable resource for the research community. At our last in-person meeting, we together came up with a prioritized list of activities that as a network we were going to work on. And some of those are listed here, including developing participant-centric educational materials about cancer genomics and communication materials, identifying optimal engagement strategies through the conduct of appropriate research, extending the definition of participant engagement to the entire research process and identify meaningful measures of engagement, and others, which are listed below. And the network has already started to work on these issues together, and we have a paper in press in JAMA Health Forum that discusses a framework for engaging diverse participants in genetics and genomics research. Now, for the last few minutes, I just want to focus on one of these projects, our project called Count Me In, which is focusing on osteosarcoma and leiomyosarcoma. This is part of a larger initiative called Count Me In, which enables patients with any type of cancer across the US or Canada to participate in genomics research. Leiomyosarcoma and osteosarcoma are both rare cancers, each with approximately 2,000 or fewer cases per year in the United States. There's been a minimal improvement in survival or treatment-related morbidity in osteosarcoma and leiomyosarcoma, largely due to insufficient characterization of the genomic landscape of these two cancers. And thus, there's an urgent need to generate a large shared database of clinical and genomic data in osteosarcoma and LMS. This has been challenging to do to date, mainly because of the rarity of these tumor types and other challenges in patient recruitment and the genomic characterization of these tumors. And so the goal of the Count Me In PE-CGS project is to engage patients with OS and LMS across the US and Canada, and together generate a large publicly available database of clinical, genomic, molecular, and patient-reported data. And we do this through an online portal at joincountmein.org, and we ask patients to sign up, to identify themselves and sign up and share their information, share their samples, share their experiences, and share their voices with the projects to help accelerate this work. And you can see in this project, as well as through other Count Me In projects, we now have participants in these projects across the US and Canada who are contributing their information to help accelerate research. Now, you're going to hear more about the osteosarcoma and LMS project from Maeve Smart, one of our participants, in just a few minutes. But I'll just highlight that the patient engagement and patient participation, and really patient partnership, is one of the most important aspects of this project. And patients and participants have been involved from the very beginning, from the conception to the design, to the implementation, and to the ongoing research. And you'll hear more details about that in just a second. So I will close with that. I want to thank our whole PE-CGS Center team, as well as the other PE-CGS centers and the NCI for all their support. Thanks very much.
Hi, everyone. Thank you so much for inviting me to share today. My name is Maeve Smart. I'm a senior at Northeastern University in Boston, studying health science on a pre-med track. I'm very grateful to also have had the opportunity to work in the Janeway Genomics Lab at Dana-Farber Cancer Institute under the mentorship of Dr. Katie Janeway while I was at Northeastern. Through additional work with an osteosarcoma advocacy organization, I was also lucky to work on the Broad Institute's osteosarcoma team, in which we helped inform the end user's view of the osteosarcoma project, and I've additionally participated in patient recruitment videos. I wanted to start by sharing just a bit more about my background before circling back to the Count Me In project. Back in 2011, when I was 11 years old, I really loved to play basketball and soccer. Towards the end of 6th grade, though, I started to have some pain in my proximal right tibia, and within a week, I entered Memorial Sloan Kettering Cancer Center in Manhattan for the first time. I did not even realize as I had walked the halls of Sloan for the first time that I was inside of a cancer hospital. I knew I was having a biopsy, I had heard the word osteosarcoma thrown around, and I knew the doctors were hoping it was a benign tumor instead. I had no idea, though, that any of these words were even related to cancer, just as no 11-year-old should. And no one in my family had ever had cancer before me, so this world was new to us all. While the diagnosis was a shock, things moved pretty quickly from there on out. Soon after, I began chemotherapy on a MAT protocol, which includes cisplatin, high-dose methotrexate, and doxorubicin. My prognosis was pretty good. The tumor was small and localized and hadn't yet spread to my lungs, where osteosarcoma most often metastasizes, and limb salvage surgery was still possible. After 3 months of chemotherapy, I had a 16-hour vascularized fibular graft surgery to remove the tumor and replace the tibia in my right leg with the fibula for my left leg. After a 17-day stay in the hospital post-op, it was straight back to chemo. The thing about osteosarcoma is that treatment has not changed in over 4 decades, and it's brutal for every single child. These drugs are so toxic that they can cause hearing loss, neurotoxicity, heart damage, and even secondary cancers later in life. But I handled them well for the most part, all things considered. I stopped treatment a bit early due to a case of pneumonia and septic shock that landed me in the PICU for 17 days, but my treatment team was still optimistic I would be okay, even with admitting that last cycle of methotrexate. 3 years out from my original diagnosis, I was still unable to walk, and it was at a post-op appointment for yet another revision surgery that it was first suspected my cancer returned. A biopsy soon after confirmed a local recurrence. This time around, my prognosis was much more uncertain. Even with the prescribed treatment plan of an above-knee amputation and more chemo, this time ifosfamide and etoposide. Unlike the first time, I now understood what my diagnosis meant and the pain that comes along with it. To say that I was devastated wouldn't do it justice. While my body responded well to MAP up until the end, the side effects from my new treatment plan were brutal. Despite Sloan being a pediatric day hospital, meaning you were treated on an outpatient basis, I was constantly hospitalized. I had intense bouts of mucositis, developed a bowel obstruction, and my kidney function was declining. My body clearly had had enough. The difficult decision to withdraw chemo early was made, but scan by scan, the uncertainty of my future slipped away. For some reason, it seemed that the amputation and 3 cycles of chemo I received was enough to save me. I slowly regained strength, finally returned to school full-time in the 11th grade after being diagnosed in the 6th grade originally, and graduated on time with my peers and moved to Boston for college. My journey to becoming involved in research is a little windy. For a bit there, I thought that I wanted absolutely nothing to do with osteosarcoma. I gave engineering a try, but slowly but surely realized that osteosarcoma advocacy, research, and patient engagement was undeniably my passion. I started out volunteering in a one-on-one matching program with newly diagnosed osteosarcoma patients and became more and more involved in patient advocacy as I joined the MIB Agents Junior Board. It was also while attending their annual Factor Conference, which is an annual meeting that brings together the leading researchers, clinicians, surgeons, and patient families, as well as osteosarcoma survivors, that I was first introduced to Dr. Katie Janeway and Dr. Corey Painter, who actually launched the Count Me In Osteosarcoma Project at the 2020 Factor Conference. What makes Count Me In so important to me is that it empowers patients to participate and help change the future course of their cancer. I have not met a single osteosarcoma patient that is not eager to do anything they can if it means that future kids will have kinder treatment options. So this project really resonated with our entire community. Count Me In has created a partnership between patients and researchers to gain new insights into how our tumors develop, what the genetic drivers of disease may be, the overall genomic landscape of these cancers, and why they are often treatment resistant. I am one of the lucky ones. Sure, 7 years out, I still think about my treatment every single day, and I'll live the rest of my life with a disability because of my cancer. But for some reason, I survived, even when my treatment team wasn't so sure that I would. And that is the norm for pediatric cancers. They are so often underfunded and neglected, leaving kids with limited options when standard therapies fail. But Count Me In changed course. They prioritize patients like me, as well as our siblings, parents, grandparents, and friends. They saw the strength of the osteosarcoma community and tapped into that. This project collects de-identified genomic, clinical, and patient-reported data, making all of the above freely available to the global biomedical community, so that we may move towards long overdue progress together. Additionally, a return of genomic results to patients that opt in is also currently being developed for osteosarcoma and leiomyosarcoma, so researchers and patients have the opportunity to learn together. Through the collaboration of Count Me In with cancer advocacy groups, which was in my case MIB agents, I have found hope in learning the important role that genomics plays in progressing how we treat cancer, and I'm very grateful for the work of everyone involved in these projects. Thank you so much for taking a few minutes to listen to my story and engaging with patients as a part of your research. Together, we can make it better. Thank you.
So we will close out this session with some concluding remarks, highlighting some of the themes that we heard across the different programs. In this session, we heard from several of the Network for Direct Patient Engagement programs. These programs highlighted for us what we see as the value and potential of these patient-researcher partnerships. This value includes learning how to do these studies better, and also examples of work that could not be accomplished without these partnerships. These programs are also working on novel ways of engaging with patients and medical providers in research. They support bidirectional communication and reciprocal relationships between researchers, providers, patients, and participants. And the lessons learned from these programs could inform future studies. And with that, I will conclude by thanking all of our presenters for their fantastic presentations, and thanks to all of you for attending our session.